Weight management research has historically focused on macronutrient ratios, caloric density, and satiety mechanisms, while treating food primarily as a delivery vehicle for calories and nutrients. This framework has produced useful insights about energy balance and appetite regulation, but has increasingly been recognized as incomplete. The human digestive system is not a sterile environment where food is mechanically broken down and absorbed. It is a complex ecosystem inhabited by trillions of microorganisms that influence digestion, metabolism, immune function, and even behavior through mechanisms that are still being mapped. Recent research has begun examining whether the microorganisms living on and in food itself, not just the established gut microbiome, might influence health outcomes. A study published in Nutrition Research examined the relationship between consumption of foods estimated to contain live microbes and various health markers in Australian adults. The findings suggest that people who eat more foods rich in live microorganisms have lower body weight, smaller waist circumference, better metabolic markers, and more favorable cholesterol profiles compared to those who consume fewer of these foods. This represents a shift in thinking about food quality. The nutritional value of yogurt has traditionally been attributed to its protein, calcium, and vitamin content. The new research suggests that the live bacteria cultures in yogurt may contribute to its health effects, independent of these conventional nutrients. Similarly, raw fruits and vegetables may provide benefits beyond their fiber and phytonutrient content through the diverse microbial communities that naturally inhabit their surfaces. The study design and methodology. Researchers at the University of Newcastle constructed a database categorizing more than 200 commonly consumed Australian foods by their estimated live microbe content. Foods were classified into 3 categories: low, medium, and high live microbe content. The low category included foods that undergo processing that kills microorganisms, cooked foods, canned products, and pasteurized items. The medium category consisted of raw, unpeeled fruits and vegetables that naturally harbor microbial communities on their surfaces. The high category included fermented foods where live microbial cultures remain viable, primarily yogurt with live active cultures. Researchers applied this database to dietary data from 58 healthy adults who completed food frequency questionnaires, reporting what they had eaten over the previous 3 to 6 months. The team also collected health measurements including body weight, BMI, waist circumference, blood pressure, fasting blood glucose and insulin, cholesterol levels, and inflammatory markers. Because relatively few foods contain high levels of live microbes, researchers combined the medium and high categories for statistical analysis. They used regression models adjusted for gender, smoking status, and total energy intake. Key findings on weight and metabolic health. People who reported eating more foods from the medium to high live microbe category had significantly lower body weight, BMI, and waist circumference compared to those consuming fewer of these foods. The differences were meaningful even after accounting for total caloric intake, suggesting that the effect was not simply due to these foods being lower in calories or higher in fiber. Fasting insulin levels were also lower in people who ate more live microbe-rich foods. Insulin is released in response to rising blood sugar and facilitates glucose uptake into cells. Chronically elevated insulin, even when fasting blood sugar remains normal, indicates insulin resistance. Cells are not responding efficiently to insulin signals, requiring the pancreas to produce more. Insulin resistance precedes type 2 diabetes development and is associated with increased cardiovascular disease risk and difficulty losing weight. HDL cholesterol levels were higher in people consuming more live microbe-rich foods. HDL cholesterol transports cholesterol from peripheral tissues back to the liver for excretion, effectively removing excess cholesterol from the bloodstream. Higher HDL levels correlate with lower cardiovascular disease risk. The relationship is strong enough that HDL is sometimes called good cholesterol, though this simplification obscures the complex role HDL particles play in lipid metabolism. The researchers also found associations between live microbe-rich food consumption and lower levels of inflammatory markers including interleukin-6 and C-reactive protein. Both are produced in response to inflammation, and elevated levels predict increased risk of cardiovascular disease, diabetes, and other chronic conditions. However, these associations did not remain statistically significant after adjusting for additional variables, suggesting they may be indirect effects or require larger sample sizes to detect reliably. Interestingly, the study found no significant relationships between live microbe intake and fasting blood glucose, total cholesterol, LDL cholesterol, triglycerides, or tumor necrosis factor alpha, another inflammatory marker. This pattern of results suggests that live microbe consumption may influence specific metabolic pathways related to insulin sensitivity and lipid metabolism, while not affecting all aspects of metabolic health uniformly. Limitations and interpretive caution. The study design limits the conclusions that can be drawn. As a cross-sectional study, it captured a snapshot in time showing associations between live microbe-rich food consumption and health markers, but cannot establish causation. People who eat more yogurt and raw produce may differ from those who do not in ways beyond just their microbe intake. They might exercise more, smoke less, or have higher health consciousness generally. The statistical adjustments account for some of these factors, but cannot eliminate all potential confounding. The sample size of 58 participants is relatively small for detecting modest effects and establishing robust associations. Larger studies would provide more reliable estimates of effect sizes and allow examination of whether relationships differ across subgroups defined by age, sex, or baseline health status. Dietary intake was self-reported through food frequency questionnaires, which are subject to recall bias and measurement error. People may misremember what they ate or provide socially desirable responses rather than accurate reports. While food frequency questionnaires are standard tools in nutritional epidemiology, they introduce uncertainty into the data. The study population was predominantly white women from a specific geographic region, limiting generalizability to other populations with different dietary patterns, genetic backgrounds, or environmental exposures. Practical applications and food choices. Despite limitations, the findings align with growing evidence that live microbe-rich foods support metabolic health. The practical implications focus on incorporating these foods into regular eating patterns, rather than requiring dramatic dietary overhaul. Yogurt with live active cultures represents the most concentrated source of live microbes in typical Western diets. Choosing yogurt varieties that list live cultures on the label and consuming it regularly as part of breakfast, snacks, or meal components provides a straightforward way to increase live microbe intake. Greek yogurt offers higher protein content while maintaining live cultures. Raw fruits and vegetables contribute both conventional nutrients and surface microbes. Eating produce raw when appropriate, leaving edible peels intact, and including fresh salads regularly increases exposure to diverse microbial communities. The variety of produce consumed matters because different fruits and vegetables harbor different microbial populations. Fermented foods beyond yogurt, including kefir, sauerkraut, kimchi, and miso, provide additional sources of live microbes. These foods were not extensively represented in the study, but contain concentrated populations of bacteria and yeasts that survive through fermentation and storage. The research suggests that food choices matter not just for their nutrient content, but for the living organisms they contain. This represents an expansion of how nutritional quality is understood and provides additional rationale for dietary patterns that emphasize minimally processed foods, fresh produce, and fermented products.